afternoon, everyone, and welcome to today's Public Health Ontario's Rounds presentation on laboratory testing methods and applications in bacterial enteric case and outbreak investigations. My name is Christine Navarro, and I'm a public health physician in communicable disease control at Public Health Ontario, and I have the pleasure of moderating today's session. Before we begin, I would like to start with a land acknowledgement. Today's rounds are being presented to you from a place we call Toronto, but for thousands of years it has been the traditional land of the Huron-Wendat, the Seneca, the Haudenosaunee, and the Mississaugas of the Credit. This land was covered by Treaty 13, signed by the Mississaugas of the Credit, while the province that we serve is covered by more than 40 treaties and other agreements, as well as unceded territory. Ontario has been and is still the home to many diverse Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work on this land. We acknowledge this as part of Public Health Ontario's commitment to truth and reconciliation. Next, I'll mention a few housekeeping items. The chat pod has been disabled to limit any distractions during the presentation. Please use the Q&A pod if you have questions during the session. A discussion and question period will follow the presentation. If at any point during the session you experience any technical issues, please email capacitybuilding@oahpp.ca. I would like to state that as the moderator of this session, I have no conflicts of interest to declare. It is now my pleasure to introduce the speakers for today's presentation, Dr. Antoine Corbet and Christina Lee. Dr. Antoine Corbet is a medical microbiologist at Public Health Ontario, where he is the portfolio lead for parasitology, enteric bacteriology, point-of-care testing, and education. Christina Lee is a senior program specialist with PHO's Enteric, Zoonotic, and Vector-Borne Diseases Unit. She collaborates with municipal, provincial, and federal partners to investigate foodborne illness clusters and outbreaks. Prior to joining PHO, she worked as a health promotion consultant and as a public health inspector at local health units. I will hand it over now to Christina.
Thank you very much, Dr. Navarro. Welcome to today's PHO round on laboratory testing methods and application in bacterial enteric case and outbreak investigation. The laboratory testing methods that we'll be addressing today in today's presentation are culture-independent diagnostic testing, known as CIDT, and whole genome sequencing, known as WGS for short. Before we begin, I'd like to acknowledge that neither Dr. Corbet nor I have any conflicts of interest to disclose. Now, we acknowledge that sometimes navigating lab methods and results can be confusing for some. As well, there may have been staff turnover in organization, and we hope today's presentation can provide you with the clarity and equip you with information going forward. By the end of today's presentation, we hope that you'll be able to utilize terminology for different laboratory testing methods, describe the use of culture-independent diagnostic testing in Ontario laboratories for enteric bacterial detection, describe the use of whole genome sequencing for enteric bacterial outbreaks investigation and surveillance, and as well assign case classification properly based on lab results and explain the impact of CIDT on incidence of bacterial enteric diseases of public health significance. And let's go over the flow of today's presentation. We'll start with Dr. Corbet describing the increased use of CIDT, as well as the application and limitations of CIDT. He'll then turn it over to me, and I'll go over how to classify cases based on lab results and the impact of CIDT on the incidence of bacterial enteric pathogens. And I'll turn it back over to Dr. Corbet to explain what is whole genome sequencing, its clustering and visualization, and I'll go over the application of whole genome sequencing and who does what with these WGS results. So you may have a better understanding of each partner's roles and responsibility within the public health infrastructure. And with that, I turn it over to Dr. Corbet.
Wonderful. Thank you, Christina. And hi, everyone. I'm Antoine. I'm one of the microbiologists working at Public Health Ontario. So let's start first with culture-independent diagnostic tests. So stool culture has been the main method available to diagnose enteric bacterial infections over the past century. Then antigen tests such as ELISAs, lateral flow tests, became available in the 1990s, but their use has been limited by their low sensitivity. The first generations of molecular tests were developed in the early 2000s, but these could only test one bacteria at a time. So then newer generations of molecular tests have become more convenient over the past 15 years because they can now test for multiple organisms at the same time, or what we call multiplex tests. So all tests that don't use culture to diagnose an infection are called CIDTs, or culture-independent diagnostic tests. Overall, we've seen an increase in the use of CIDT methods globally. For example, most bacterial enteric cases in the United States 10 years ago were diagnosed using culture methods only, as shown by the light blue columns in the top right graph. By 2023, the trend has flipped, with only a fraction of cases diagnosed by culture only. The majority of cases are now diagnosed by CIDT instead, and 47% had either a negative culture or no culture done whatsoever. So we'll see why this trend can be an issue. So with traditional culture, the routine goal is to find 4 main organisms: Salmonella, Shigella, Campylobacter, and E. coli 0157. Most of you are likely familiar with these organisms. Depending on the clinical and epidemiological risk factors, cultures can also be used to find other organisms. And culture can take up to a week, with the report usually containing the term isolated, which means that the lab has grown a pure concentration of bacteria, which can be used for further analysis. On the other hand, culture-independent tests are often based on a PCR platform, which, depending on the kit used, may detect a wide range of organisms, including those listed in blue here. Some of these organisms are not diseases of public health significance, and their relevance is still debated. With CIDTs, results are usually available within the same day. And because the test only looks for fragments of an organism, as opposed to growing a pure organism on a culture plate, the report will usually have the term detected instead of isolated. The lack of a concentrated amount of bacteria growing on a culture plate means that further testing is usually not possible by CIDT alone, unless the lab decides to culture the specimen after a positive CIDT test. If culture is only performed when a CIDT result is positive, we call that a reflex culture. In practice, reflex cultures are not always performed by all labs or for all positive CIDT results. Compared to culture, molecular CIDT panels are simpler, faster, more tolerant of specimen degradation, and usually have increased sensitivity. However, CIDTs can be costly, can generate false positive results, and may detect organisms of unclear clinical or public health significance. Some organisms or species might not be distinguishable when using CIDT panels, and CIDTs are also unable to provide typing or susceptibility results. CIDTs may also detect residual organism fragments in the sample, which doesn't necessarily mean that a patient has an ongoing infection. So these limitations highlight the importance of maintaining capacity for culture across labs in Ontario. For example, some commercial CIDT panels cannot distinguish between Shigella and entero-invasive E. coli. While Shigella may require treatment and is a disease of public health significance, the significance of entero-invasive E. coli is less established overall. So labs using CIDTs cannot distinguish between the two and may end up reporting results as Shigella/EIC detected. To distinguish between the two, labs would either need to use a different molecular test or would need to perform reflex culture on the specimen after the positive CIDT result. Similarly, some CIDT panels might not distinguish between Vibrio cholerae and non-cholera Vibrio species, such as Vibrio parahemolyticus. So although cholera and non-cholera vibriosis have different implications, CIDT labs might report results simply as Vibrio species detected. These labs would then have to use a different molecular test or do reflex culture to confirm the species. And again, the same issue applies here with typhoidal and non-typhoidal Salmonella infections, which both have different implications, but which might not be distinguishable readily by CIDT. CIDT labs would report the result as Salmonella species detected and would have to do reflex culture in order to isolate the organism in sufficient concentration to allow for serotyping. As some of you know, we're also seeing a rise in antibiotic-resistant bacterial infections, such as Shigella sonnei, shown here. Unfortunately, the only way to test for enteric bacterial resistance is through culture. So if labs only perform CIDTs without reflex culture, we may lose the ability to track resistant trends over time. Another limitation of CIDTs is the fact that they can detect dead organisms or residual fragments. These dead organism fragments may persist even after the infection has resolved. For example, this graph from a study in Alberta shows that the stools of individuals with Shiga toxin-producing E. coli may remain PCR positive for 2 to 7 more days compared to traditional culture. Although CIDT panels are quick and have a great negative predictive value, the relevance of a positive CIDT result but negative culture result is less known. To circumvent these limitations, all labs in Ontario are expected to perform reflex cultures locally following a positive CIDT result. But the reflex cultures might not always turn out to be positive. For instance, while up to 80% of Salmonella CIDT-positive specimens might show growth of the organism on reflex culture, that number may fall well below 40% for organisms such as Campylobacter, Vibrio, and Yersinia. Potential reasons why a sample might be CIDT positive, but culture negative, include a low initial concentration of the organism, a loss of organism viability, an overgrowth of normal gut flora, a high culture requirement for some strains, residual fragments detected, or a random false positive signal. Christina will then provide us some examples on how this can impact public health case management.
Thank you. So it's important to understand how to classify cases based on the lab results, be it CIDT and culture. And this table provides an overview. And before I dive into the table, I want to provide some of the keywords that Dr. Corbett has already mentioned. When a lab report have keywords like real-time PCR, PCR, NAT, or EIA, the method that was used is CIDT, and the case is a probable case. And as for Shiga toxin producing E. coli cases, if the keywords are STEC1 or STEC2, that's also detected by CIDT method. Compared to when a lab report has the keywords such as culture and isolated, it means that culture was performed, and if the culture is positive, it's a confirmed case. Now let's go over the table together. When a clinical specimen is collected and it undergoes CIDT testing and it's detected, meaning positive, it starts off as a probable case. If the culture is performed, if the culture is performed and is positive, it goes from a probable case to a confirmed case. However, if the specimen is not cultured or it's culture negative, it remains as a probable case. Now, if a clinical specimen is submitted to a lab where they either do not do CIDT or the pathogen is not detected by CIDT, in which case, and it goes straight to culture, it becomes a confirmed case. However, if the specimen is neither not tested by CIDT and also either not isolated by culture or culture is not performed, in which case there's no laboratory confirmation that this case is a lab confirmed case. So when you look at these lab reports, it's important to bear in mind about the culture recovery yield that Dr. Colbert has mentioned. Next, let's go over some examples of how to enter these results into IFAS, as they may vary by pathogen. So for STEC cases, if it's detected by CIDT, it's a probable STEC case. If it's isolated by culture, it's a confirmed STEC case. However, if you receive a lab report where it says detect Salmonella by CIDT, please enter it as a probable Salmonellosis case in IFAS. And upon isolation by culture of a non-typhoidal Salmonella, please update the probable case to a confirmed case. And if the culture isolates Salmonella typhi or paratyphi, please close the Salmonella case as does not meet and create it as a new typhoid fever or paratyphoid fever case. And lastly, for lab results indicating that Vibrio species is detected by CIDT, if it's reported by the laboratory, consider classifying as a confirmed food poisoning case. If the culture comes back positive for Vibrio cholerae, you may update that case to a confirmed cholera case and close the food poisoning case as does not meet. And lastly, I'd like to go over the incidence of bacterial enteric pathogens due to the impact of CIDT. We selected SCHECK here as an example because the use of the CIDT for SCHECK has been established longer compared to other bacterial enteric pathogens. And as you can see, the line on the top in blue has a downward trend for 0157 E. coli. And this in part may be due to the lack of reflex culture or the culture yield. And when you look at the green line on this diagram, you notice there's a slight increase of non-0157 E. coli between 2018 and 2019, and also when comparing 2018 to 2023. And this may be due to the greater sensitivity of CIDT. And lastly, the incidence of probable cases have remained fairly stable over the years. And now I'd like to pass it back to Dr. Corbert for whole genome sequencing.
Wonderful, thank you, Christina. So whole genome sequencing is another lab method that may be confusing for some. So we'll explore its use case today for enteric bacterial testing. So typing means characterizing a bacteria beyond the genus or the species level into separate unique strains or variants. Since the 1960s, the main typing process for enteric bacteria has been serotyping, where we identify specific antigens on the surface of the bacteria and give the antigen profile a code name like typhi or 0157H7. Other methods, such as plasmid profiling, phage typing, and pulse-field gel electrophoresis, were used until whole genome sequencing became standard in 2017. And since the early 2000s, all public health labs in Canada and the US have joined force to do routine typing of key enteric organisms to proactively detect outbreaks. That includes Salmonella, Shigella, Shiga toxin-producing E. coli, and Listeria. And that group of public health labs is called the PulseNet network. So what is whole genome sequencing? Essentially, whole genome sequencing is the process of documenting the full DNA sequence of an organism. At this time, we can only do sequencing if we have a purified concentration of bacteria from a culture plate. So sequencing directly from specimen for enteric pathogens is not available at this time. The DNA sequence can be looked at the level of each individual letter, called nucleotides, or can be looked at a broader DNA segment scale, and each of the DNA segments are called a locus or a gene. There are different ways to compare between two different bacterial sequences, but here are some common ones. One of them is to go through each DNA letter one by one and count the number of mismatched letters, which we call single nucleotide variants, or SNVs. You might have also heard the term polymorphism, or SNPs. So in this example, we can say that there are 5 mismatched letters, or SNVs, between isolate A and isolate B. A second approach would be to go through DNA segments and count the number of mismatched segments, which we call alleles. So in the same example, although there are 5 mismatched letters, their position would only impact 2 out of 3 genes. So we would say here that there are 2 allele differences between isolate A and isolate B. If comparing more than 2 sequences, it's sometimes easier to visualize the differences on an evolutionary tree, or what we call a dendrogram. There are a wide range of ways to create dendrograms, but here's a standard approach that the Canadian PulseNet network uses for enteric pathogens. each box here represents an isolate, or if you will, quote unquote, the leaves of the tree, each represent an isolate. The isolates are linked by horizontal lines, or quote unquote branches, and the length of the branches represents the number of differences between an isolate and the next closest group on the tree. To make it simple to understand, we can look at the following example here. So let's say that you have a list of 4 cities and would like to compare how cities cluster to each other. In this example, let's say that Toronto and Charlotte are the 2 closest cities among our group at 9,500 kilometers apart. So that will be our first grouping. The next closest neighbor after that would be Thunder Bay, which would be 12,700 kilometers away from the midpoint between Toronto and Charlotte. The last neighbor on the list would be Rimouski, which would be 13,600 kilometers away from the average point between Thunder Bay, Toronto, and Charlotte combined. That being said, clusters can change drastically when you add new data points. So for instance, if we were to take the same 4 cities, but add a 5th city, Sault Ste. Marie, now the dynamic has changed because the 2 closest cities to one another would now be Thunder Bay and Sault Ste. Marie at 4,200 kilometers apart. Then the next closest city after that would be Toronto, which would be 7,200 kilometers away from the midpoint of Thunder Bay and Sault Ste. Marie. Then we have Rimouski, which would be 11,900 kilometers away from the combined group of Thunder Bay, Sault Ste. Marie, and Toronto. And lastly, Charlotte would now be the farthest city from the rest of the group at 14,300 kilometers from the meeting point between Thunder Bay, Sault Ste. Marie, Toronto, and Rimouski. So this highlights the fact that cluster pairing is very relative and depends on the samples tested. Finding hidden links may shift and reframe the clusters into new groups over time. To standardize interpretations, PulseNet Canada has established initial cluster screening cutoffs based on the number of allele differences. Essentially, when isolates are grouped together with 10 allele differences or less, it's suspicious that they could be originating from the same outbreak source and would require further public health investigation. Once an initial cluster is screened positive by whole genome sequencing, PulseNet Canada assigns a cluster with a unique code number that specifies when the cluster was initially recognized, the organism involved, the province where the cluster was first recognized, and whether or not other jurisdictions are also linked. So for example, cluster code 1906 TH WGS 1QC MP refers to a Samuel-Thompson cluster that was initially recognized in June 2019 in Quebec, but that had cases in other jurisdictions. An important message here is that epidemiological and food safety information is essential regardless of preliminary WGS clustering. So for example, a source can be contaminated by multiple bacterial strains, which won't necessarily show up as the same cluster by whole genome sequencing, but would still be related due to the same exposure source. Or for example, there could be a highly mutating strain that expands the WGS cluster beyond 10 alleles while still coming from the same exposure source. And alternatively, there could be a highly conserved strain in the environment with barely any DNA mutations over the past years, but that led to multiple exposure events that were independent. So as my colleague Christina will show next, genome sequencing is increasingly valuable, but it's unfortunately just one piece of the puzzle when investigating outbreaks.
Thank you very much. Now, let's understand how to apply whole genome sequencing for outbreak detection and investigation. Starting with outbreak detection, we have 2 images here. On the left, we have an outbreak that started off with multiple complaints or for a common event or food premise, or as a report of an outbreak in an institutional setting. In which case, this this scenario, an outbreak is detected before a clinical specimen is collected. The WGS is used here to strengthen the epi evidence and the finding from the food safety investigation for a point source event. Now, on the right, you have another image that starts off with increase of cases for a particular organism when the epi data is still pending or only partially available, thus lacking a strong signal to indicate a commonality. WGS is then able to pick up an outbreak by selecting the cases that are genetically related from those that were genetically unrelated. And this allows for the detection of both point source outbreak and also outbreaks where the source is geographically dispersed. Example of such an outbreak is the Salmonella infantis outbreak that was linked to unlicensed shredded pork rind and pork skin in 2021. Next, we have WGS is able to help rule out cases during an outbreak investigation. Here is a dendrogram of a multi-provincial Salmonella infantis cluster. As you can see, the outbreak isolates belong to that of cases and food specimens are in green and highlighted in the middle of the page. During the same time of the outbreak, we have 3 cases that came in that were assigned to the same cluster code. However, based on the epidemiological evidence, the traceback activity, we noted that these cases did not eat pork or they ate pork from an approved source. In consultation with PHO Laboratory, National Microbiology Lab, and FAC, we were able to rule out these cases as not being part of the outbreak. Based on the epi evidence, the traceback activities, and the WGS results. This example highlights how useful having a dendrogram is, particularly when you have an outlier case assigned to the outbreak's cluster code, where the epi does not match the confirmed source of an outbreak. It also highlights the need to interpret WGS results in combination with epi evidence and food safety investigation findings. And in summary, WGS can help with outbreak detection, case finding, particularly for cases outside of the affected jurisdiction, rule out cases during investigation, and also strengthen existing epi data and food safety investigation findings. Next, I want to talk about how sometimes there are variants from the rules, and follow up to what Dr. Colbert has mentioned about the 10 allele starting point. Here is a representative set of data that FAC and MNL use to validate whole genome sequencing for Salmonella outbreaks. Of the 12 outbreaks, you can see that 7 of them have an allele range of 10 or less, while 5 of the 12 have a larger allele range. And if you look at the source for these outbreaks with larger allele range, you noted that the source of them would suggest it may be due to polyclonal contamination, as is due to reptiles, feeder mice, or frozen breaded chicken products that are grounded up. Similarly, FAC also did a verification for E. coli outbreaks using 10 outbreaks. You can see out of the 10, 7 of them have an allele range of 10 or less, and 3 of the 10 have a larger allele range. When you look at the source of this, such as frozen beef burgers and ground beef, you can also imagine that it may have been due to polyclonal contamination as well. So in short, within the 10 allele range, while it's validated and established by FAC and MNL, and it's a good starting point, it is important to bear in mind that WGS results is meant to be interpreted in combination with epi data and also investigation findings from food safety investigations. Now let's take a step back and look at the bigger picture and understand the flow of information between public health partners and how we work together. We want to recognize the important roles a health unit plays in collecting epi data from the initial case investigation and the inspection of implicated food premises, including the collection of food specimens and environmental swabs. Together, these 3 streams of evidence are crucial to an enteric outbreak investigation and allow the system to work as a whole. Now, let's talk about who does what with these results. PHAC would assess multi-provincial WGS clusters, and Public Health Ontario will assess Ontario-only WGS clusters. When Ontario-only WGS clusters have cases that are exclusive to one health unit, we'll notify that health unit accordingly, and they in turn will be the lead to assess these clusters and determine if investigation is warranted. Sometimes there are also subclusters within multi-provincial or Ontario-only WGS clusters. And these ones tend to be detected either one, when health units notify us of local outbreaks, we monitor for whole genome sequencing results, and we note that they are a subcluster within an existing cluster by requesting a dendrogram. Or two, the other way we notice it is when we notice an increase of cases within an existing cluster code. If the cases within the subclusters belongs to Ontario, in which case PHO will assess this cluster. However, if the cases are isolated in one health unit, the health unit will lead the assessment of that cluster code. And if the cases are dispersed across Canada, then PHAC will assess and lead that assessment. Now, in terms of volume of results, it's important to have an idea of how the amount of results are coming in. Here, we'll give you a sense of the number of WGS cluster code by pathogen and by jurisdiction. As you can see, there's a lot more Salmonella clusters than there are for Aztec, Listeria, and Shigella combined. And a lot of these clusters are at the level of 2 or 3 cases. Let's turn our attention into understanding how we go about assessing these clusters, and hope it'll give you some insights into the thorough process behind our examination. So we use 3 streams of evidence or information. The first one starts with PHAC and how they go about classifying WGS clusters. As you can see, they have 7 classifications for WGS clusters. They are poultry, travel, sexual transmission, zoonotic, homeless, underhouse, other, and unknown. We want to highlight the role of the health units in conducting their case investigation, and the more complete the data set is in terms of risk factors from their initial case interview, the more useful it is. This data helps the federal, provincial, and local partners in identifying a commonality, such as travel abroad for a Salmonella cluster or a common food premise, at a local level. Also allow us to classify cases properly into these categories and to note when things are happening unexpectedly. It also allows us to target our resources into the clusters of unknown classification. As you can see here, a majority of the Aztec, Listeria, and Salmonella clusters are classified as unknown and may warrant further assessment and investigation. The second stream of evidence comes from the health unit's interview. So as you can see, under epidemiological evidence, we'll give more weight when the cases are coming in fast and furious, or perhaps it's for an organism with more severe outcome, and also for a particular demographic profile. We also consider things like laboratory evidence, whether or not this is a rare serotype, if the WGS results is available yet. And also under food evidence, we'll also evaluate whether or not there are any matches to non-clinical specimens already there that may provide a hypothesis. Upon completion of assessment, we'll determine which cluster warrants further investigation or data, at which point we'll notify health units of WGS results, and we'll do so in 2 ways. The first is by email, when a WGS cluster belongs to a single health unit. The second way we notify, and the other way we also notify is when the clusters belongs to has cases across Ontario or across Canada and warrants further assessment by either PHO or PHAC. We'll also share WGS cluster code of outbreak cases for local outbreaks upon notification. And if warranted, health units may also request PHO laboratory to produce a dendrogram, and this is particularly useful when you have outlier cases in your outbreak. The second way we notify health units is when we have provincially led or PHAC-led outbreaks investigation. in which case we'll list the IFAS case ID for all the outbreak cases under Ontario Outbreak Centre of the Canadian Network for Public Health Intelligence website. There's a post for each of the active outbreak. In order to gain access to the website, you will need an account, and instructions on how to do so can be found in the weekly ESD and monitor situation document. Dendrograms that are produced for these outbreaks may also be posted there. I also want to take this opportunity to highlight that in a PHO-led investigation, the activation of an Ontario Outbreak Investigation Coordinating Committee under the Ontario Foodborne Illness Response Protocol is a separate event from the declaration of an outbreak. Sometimes it does not move forward when the cases dries up or there's no lead. Similarly, the deactivation of an on-OICC is a separate event from the declaration of the outbreak being over. Now, as we come to a close to today's round, I'd like to give you one last example of how CIDT and WGS can work together for outbreak detection and investigation. In March this year, PHO observed 4 cases of E. coli in a single health unit, all having a common ethnicity. 3 of the cases were O157H7, and 1 was a probable case with STEC2 gene isolated. Public Health Ontario notified the affected health units, and they noted that 3 of the 4 cases reported dining at the common food premise. Later on, 2 additional cases were added, and upon further investigation by both PHO and the affected health units, we noted that all 6 cases actually reported eating at the common food premise. 5 of them were O157H7. WGS were then available to confirm the linkage, and there was 1 probable case. This highlights that sometimes CIDT can support and strengthen outbreak investigation when paired with culture and whole genome sequencing results and timely and good epi data. And it also shows how WGS can confirm linkages and assist with case finding when the cases fall outside of a health unit. And in summary, CIDT are a useful laboratory method for diagnostic purposes. As community and hospital lab use CIDT more, there's also expectation that they perform reflex culture following a positive CIDT results. And if there's no reflex culture or the culture is negative, cases of bacterial enteric pathogen will remain as probable and will not be able to determine if the case is part of an outbreak. And WGS can offer greater accuracy in determining and identifying potential genetic relatedness amongst bacterial isolates. An outbreak may be single genetic strain or can have multiple genetic strains, which may or may not suggest different transmission events. And WGS need to be interpreted with epi data and also food safety investigation data. Outbreaks may be due to a single or polyclonal strain or serotype, and the allele range and the relative distance may vary by outbreak and over time. And lastly, collaboration amongst partner and good data quality can inform public health action. And with that, thank you for attending today's PHO round. I'll turn it back over to Dr. Navarro.
Thank you, Antoine and Christina. We will now move on to the Q&A segment of this event to address some of the questions from our audience. Please continue to enter your questions into the Q&A pod regarding the topic of today if you have not already had the opportunity to do so. And I do have some questions here in the chat pod that are ready for you. The first one I am going to send over to Christina. The question is, if you receive a positive culture for Salmonella species, should this result be entered as confirmed Salmonella or probable until serotyping is completed to confirm it is non-typhoidal?
Hi, I'm afraid I need to turn that question to Dr. Corbert. I don't think I've ever seen a culture result without the species level.
No, it's a good point. There's maybe a bit of a gap between the culture result identifying the salmonella organism to the serotyping that describes whether if it's a typhi or an enteritidis or typhimurium and so forth. Um, with that gap in mind, most labs will report it once the serotyping is available, but if not, then there's a bit dependent on what the clinical and epidemiological information provides. In most instances, they would probably fall within the salmonellosis case, but then if a typhi or a paratyphi result comes back, similar to when we do the CIDT, then the case would have to be reshifted to a typhi or a paratyphi case.
Thank you. This one should go to Antoine. Is the use of MALDI-TOF considered a CIDT? And maybe you could tell us what MALDI-TOF is first.
Great question, indeed. So the microbiology labs have, for the most part, moved towards the equipment called MALDI-TOF, which is essentially a way to analyze the proteins that are found in the bacteria itself. And with that analysis, it's able to identify the organism, usually at the species level, if not at the genus level. So it is a new method to identify organisms by looking at the proteins within the organism. That being said, most MALDI-TOF instruments require to have culture first. So if you see a result that says identified by MALDI, it is a fair assumption to think that the organism was cultured first. And then once the culture is completed, that's when the MALDI instrument is being used to identify the organism within the culture itself. So at this point, it wouldn't be considered a CIDT, but it would be considered a method to identify the organism by culture.
Here's another lab-related question. So CIDT used to identify STEX1, STEX2 positive samples without culture is a probable E. coli case. But how does exclusion apply if you don't get a positive culture, since CIDT positive does not indicate actual infection? Actually, this may be a question for Christina as well, since it applies to exclusions.
Sure. Happy to go first. Unless Krishna, you
Sure.
want to go ahead?
Go
Yeah,
ahead.
that is a great question as well. So as we mentioned, there is potentially persistence of the fragments of DNA, for example, for the STEC1, STEC2 genes. In that case, we had a few questions that are similar to this. It used to be more on a case-by-case basis to do a risk assessment to understand if the individual is still shedding the organism or not, based on the symptoms, based on the temporality of the positive results. Different jurisdictions have approached it differently. I don't know, Christina, if you have anything else to add.
I think it needs to be interpreted with EPI. Currently, probable STEC case requires exclusion criteria. That is something that is on our radar, and we hope to address it in the future.
I have another question related to exclusion criteria. Maybe you've just answered that, Christina, but what are the case management exclusion recommendations for probable cases detected by CIDT, for example, Shigella and typhoid?
The exclusion criteria for a probable case is the same as a confirmed case.
Here's another question, and I think this is for Christina. What are the expectations of PHUs or public health units to follow up with labs on cases reported positive on CIDT, as public health units do not receive the negative culture reports from labs?
Excellent question. Currently, we do not request health units to actively follow up the lab to determine if there is a culture negative results. Those are usually done when the scenario requires perhaps in the investigation of an outbreak, and we suspect the case based on demographic or the geography of the case that it may be linked to the outbreak, and that's the only circumstance when PHO will reach out to health unit and ask them to follow up on culture results.
This might be one for you. What is the harm of having CIDT as part of the confirmed case definition? It seems like we are underreporting if we are only able to classify as probable. Who would like to go first?
I'll start and perhaps you can add to it. So we are currently trying to understand the impact on CIDT on bacterial enteric pathogens. And due to the limitation of IFAS, and also the improper classification of cases right now, we're not able to get a full sense of this data. And so as such, we classify them as probable when it's detected by CIDT and confirm when it's isolated by culture so we can better understand the impact of CIDT. Do you want to add to that, Dr. Colbert?
Indeed, great points. I think overall these are questions that we're also hearing from all the different jurisdictions as well. So because of the increasing use of CIDTs, we're trying to really assess what is its impact. Because we're seeing cases that are CIDT positive but culture negative, and what does that mean from a clinical standpoint? What does that mean from a public health standpoint? Is there a risk for transmission? Is there no risk for transmission? So all of these questions are being assessed currently, but these are great questions.
Often we receive CIDT results like this. Shiga toxin producing organism, STEC, Shigella dysentery, detected, but then culture cannot be carried out. Should this be treated as a probable STEC or probable Shigella? I think you both have maybe some thoughts on that. Antoine, go ahead first.
Oh, thanks, Lucien. Indeed. So that's a question that arises often as well. So it depends on what testing is being performed. For the most part, there's one specific Shiga toxin that is STX1 that has maybe some overlap between the E. coli producing Shiga toxin isolates versus the Shigella dysentery. So both can do the STX1 positive result. So in that case, it is a bit challenging to distinguish between the two. What is useful is if there's also a Shigella result that was tested and provided at the same time. So if, for example, the lab has tested for Shigella and the Shigella is negative, then it does suggest a bit further that it could be an STEC as opposed to a Shigella. But if labs haven't performed Shigella testing, then it's a bit more challenging to interpret at this point. If the result is specifically for STX2, the STX2 is a little bit less overlapping with Shigella. So here it's a bit more intuitive to assign as a probable Shigella toxin E. coli as opposed to Shigella. I don't know, Kristen, if you want to add something.
Okay, and this is just to let the audience know if you have any questions, please put them into the Q&A pod, as we do have a few minutes left before the I have a question, if I could maybe ask Antoine. Last week there was a PHO rounds on laboratory surveillance of Neisseria gonorrhoeae. And one of the things that they're doing is that they've been able to take results from all of like the whole genome sequencing that they've done or the molecular testing that they've done the whole genome sequencing that they've done, and they've been able to sort of predict antimicrobial resistance for those results for those specimens that they're not able to do, that they don't have culture results for. Are we going to be seeing that, or is that something that will is being worked on for enteric diseases?
Well, thanks, Christine. That's an excellent point. It's something that is being investigated at the research level at this time. So it's not something that is actionable, provided for clinical management, for instance. But now that we're able to really open the book and really see what's inside the organism, the genes, the dynamics of the genes over time, we're able to really see whether a gene is predictive or not of developing resistance or showing as resistant when we test it in our hands. So that's something that we're excited to look into. But at this point, it is still a research-based approach that we're investigating. But in the near future, I think there's going to be excitement to move that forward. I would certainly say so.
Thank you so much. I think that would probably help a little bit with some of that, some of the limitations that we've been discussing in today's presentation. So at this time, I don't see any further questions in the Q&A. However, I know that at the end of your slides, you have put your contact information. So I hope that our members of our audience would feel free to contact you with any questions related to your presentation. And so perhaps I can bring this to a close. And so as we wrap up today's PHO rounds, I'd like to thank Dr. Antoine Corbet and Christina Lee for presenting. I'd also like to thank everyone who joined us for today's webinar. You can expect to receive a brief and anonymous PHO round survey for today's session. Please try to complete this to help us improve our programming. Lastly, access to past PHO presentations and to view confirmed and upcoming sessions, please visit the PHO website, head to Education and Events, and click on Presentations. Thank you and have a wonderful day.